Good afternoon, everyone. So hope you're not too tired by now with all these presentations. This is a little bit more like an informal presentation. So we're going to talk about the design challenge for this year and what is expected of you. What is the topic that we're going to be talking about? And here is the link where the presentations will be available to you. So if you have any questions or you want to review some of the topics that were talked about during the morning, you can go to here. There's a password. I think the link sent it to you by mail. So just input the password and download the presentation that you want. That's my email as well. If you got any questions during the week, maybe I can help you. If you need to, I don't know, you want some insight into some papers you could read about or something like that, send me an email. I'll try to answer it as soon as possible and as best as I can. So let's go. This is what we're going to be talking about in this presentation. We're going to talk about the objective of this design challenge, some background information on the topic, what's currently being done, and then why the use of micro and nanofluidics or nanobioengineering is good to tackle this problem. And then we're going to talk about what's the design challenge in itself, like the parameters, what's expected of you, etc. And then we're going to have some time for questions, and I'll put some selected references for you. So first of all, and this design challenge, what we want you to do is to apply the knowledge that you will be gathering throughout this week, and then to tackle a real world problem, think that you're researchers working on this topic, and then what kind of answer or what kind of platform you would develop for this particular situation, which in this case would be the purification of exosomes, isolation into specific subpopulations, and then integration with downstream genomic or proteomic analysis systems. And here we have a little picture of what are exosomes. It's an artistic representation. I don't think things look like this in real life, but colors are always helpful. Okay, so first of all, what are exosomes? So in cells, we usually have a lot of microvesicles within the cytoplasm. There are a lot of vesicles of different sizes. For some reason, researchers said that they found small vesicles, very, very small, 30 to 120 nanometers in diameter, and they said, this looks like exosomes for us. So they get together and they say, these are exosomes. I don't think there's a real reason why this particular size range, but this is the accepted definition of exosomes. So these little vesicles contain nucleic acids and proteins that are particular to the cells of origin from where these exosomes come from. And this is important because you get information from the interior of the cells if you are able to analyze the contents of these little vesicles. So it's been found that they are secreted by all cell types in culture. So whatever cell you have in culture, you analyze the medium, and then you can get a lot of these little vesicles floating around if you're able to purify them. And they're also found to be shed in particular high rates in cancer cells. So this is very important, especially for the diagnostic of cancer. If we already know that exosomes are secreted in a higher rate by these cells, then it's possible that these exosomes can be used as potential biomarkers for diagnostic of cancer, like early diagnostic of cancer, and then also for prognosis, analysis of how the disease is evolving, and maybe even for some therapeutic applications later on. And another thing that is known is that these vesicles have specific markers on their surfaces. This, for all cells, you have the lipid bilayer, the plasma membrane, and then within this membrane you have some proteins that are embedded within the membrane. Some of these are markers, and they can be targeted by antibodies. So if we know there are certain antibodies that can specifically target these markers, then we can see that it's possible to develop platforms that can target these vesicles and then probably isolate them, capture them, purify, and do some downstream analysis with them. Let's see. Okay. So here we have another picture of how scientists think nowadays that exosomes are formed within the cells. So it is thought that they have an endosomal origin. So they're generated within the cytoplasm of the cell, We have these multi-vesicle bodies in which like a lot of exosomes are created, and then they're stored like within a bigger vesicle. And then some of these bigger vesicles go through probably two pathways. One of them is a fusion with a lysosome, which would end up in the digestion of whatever is in the contents of this larger vesicle. And the other pathway would be to fuse with the actual cell membrane, and then the exosome will be expelled towards the environment of the cells. Now, it's pretty interesting that nowadays people are finding that these exosomes are not just like waste products of the cell. They're not like a garbage bag that the cells are throwing away to space or something. They found that probably these exosomes play a very important role in intercellular communication. So this is very interesting because previously we thought that only through hormones or other signalings is how cells communicated with each other. But now they see that these little packets could find their way to another cell, get internalized, and then elicit certain specific responses on the target cells. Especially for cancer, they're seeing that exosomes might play a role in, let's say, converting healthy cells into cancer cells due to the contents like nucleic acid contents or maybe some proteins that activate pathways in the target cells. So they think like the cancer cells sense they're like missionaries, and they go and just convince these other healthy cells to become cancer cells. And this is like a new pathway that's being discovered for metastasis. Previously, apparently, we knew that there were circulating tumor cells going around in the blood vessels, and then they internalize into a tissue, and then they like colonize and spread the cancer. But now it could also be due to this exosome. So if we're able to understand how this works, then maybe we're able to stop metastasis processes. And also, if we're able to understand how this intercommunication with cells work, we could also be able to use exosomes and like engineer the contents of the exosomes, send them to the cells, and make them work in particular ways that would be useful for us to treat a disease or something else. Okay, so we're talking about what they are, and the relevances of why we want to study exosomes then is this. So they reveal important information of the cell of origin. As we say, they have an endosomal origin. So whatever is contained in the exosomes is like a representation of what the cell contents will be in like in the bigger picture, right? And also very important in cancer, not only because of the contents of the cells, but we're talking about how they communicate through exosomes with other cells, and also because they're more they're producing higher rates in cancer cells. So it tells us that there are potential good biomarkers for cancer diagnostics. And these are some of the pathways or the things that people have found that exosomes are related to. Some of them is, as we were saying, like converting sort of normal cells into cancer cells. They also found to have an effect on immune system inhibition. So apparently some cancer cells send exosomes that have an effect on the immune system, which is also helpful for the spread of cancer. And they also found that they have effect on angiogenesis, which is also important for cancer growth. Also associated with fibroblasts, cancer-associated fibroblasts, and also in this epithelial to mesenchymal transition of cancer cells, which is one of the pathways by which they think cancer spreads. So normally the cancer goes through this epithelial to mesenchymal transition, and that's when the cancer cells start to invade the blood vessels. They travel around the body, and then they colonize other tissues, and that's how one of the metastasis pathways. Okay. So what is the challenge for exosome isolation? Like in the field, what are the challenges that we have? First of all, well, we said that they're very small in size, so 30 to 120 nanometers. So now we're not only in the micrometer range, we're going towards the nanometer range. So this is very, very small sizes. So fabricating tools or microfluidic platforms within this range has its own set of challenges. the smaller you go, the harder it gets to fabricate. Another thing is exosomes are highly heterogeneous, not only in their contents and nucleic acid contents and protein contents, but also on the markers that are on their surface. And this is because, the pathway by which exosomes are formed is not fully understood, but there's like a stochastic nature to it, like just like a little portion of a membrane forms an exosome, and then whatever was around it gets internalized, and whatever markers were on the surface is what the exosome will have. So there's a heterogeneity within even the exosomes that are generated in a single cell. So a single cell will not generate the same exosome all the time. So if you could analyze the exosomes that are being secreted by a cell, they will be heterogeneous. And then if you analyze the population of exosomes that are secreted by a lot of cells, you also have even a higher heterogeneity. Another challenge with this is that we want to specifically isolate them from a background of cells and biomolecules in biological fluids. So exosomes are found in biological fluids like blood, urine, cerebrospinal fluid, even saliva, whatever. In most biological fluids you will find exosomes. So if you want to isolate them from a bunch of other cells and proteins, then there's a challenge because you have to be able to separate them and then arrest them in a certain place and get rid of the rest of the background. So that's a challenge there. And also we want to differentiate also between normal versus cancer cell exosomes, for example. So we want to be able to tell that the exosomes that we are capturing or analyzing come from a cancer cell or maybe a normal cell. So because we know all cells will secrete exosomes, just by capturing them is not enough. You need to be able to tell that if they come from a healthy cell or from a cancer cell, and then you can get some more information, right? Okay, so we say it's difficult to capture them, it's difficult to purify them, but we already know some things about exosomes, and then how do we know these things? Currently, people have been using ultracentrifugation for the purification of exosomes from biological fluid samples. Ultracentrifugation is just like a normal centrifuge, just like a lot of times faster spinning. You go to around 100,000 to 120,000 Gs, so that's quite a lot. You get RPMs of very high, like higher than a F1 race car, I think. So this is quite fast. And at the end, you get a pure sample of your exosomes. But the problem with this ultracentrifugation is that it takes a lot of time. So this is an example of a procedure, like a normal procedure for exosome isolation from plasma or serum. As you can see, there are a lot of steps along the way, and some of the steps take hours. Most of these procedures are done overnight, because if you start in the morning, it will probably be done at night, and then you want to go home. You don't want to stay in the lab at 12 at night, so you let it run overnight, you go in the morning, do whatever you have to do, but it takes a lot of time. And also sometimes the resuspension and spinning, it's kind of difficult because of the size of the exosomes. So this method works. It allows to get a purified sample of your exosomes, but it's complex and it's also time consuming. So some things have been done using this purification method, but we want to move away from this so that the test can be done faster, maybe in a more easy manner, so that people can use them in certain labs without the need of complex and expensive machinery, right? Okay. So this is where micro or nanofluidics or nanobioengineering will come into place. So first of all, as it was mentioned in previous lectures in the morning, we need small tools for small particles. So if we want to manipulate small things, we want to build small tools, because otherwise it's hard to manipulate, it's hard to interact with this particle that we want to, right? You wouldn't use like a sledgehammer to just nail something. We also want to move away from the ultracentrifuge, because even though they work, they're complex, they're time consuming, and time is money, so people want to make this faster, they want to make it easier, and also people in certain labs, they want to get into using the exosomes even if they can't afford, for example, an ultracentrifuge, or maybe they don't have the space anymore or something like that. So we want to make it a little bit easier. And this will also allow for a more in-depth study of exosomes, because if you can separate them into subpopulations, maybe analyze where they come from, lyse them, make genomics, proteomics, then you can have more information on how this works. Maybe even some people are trying to studying how the exosomes interact within cells. So if you can see the cells grow them in a microfluidic channel, and then you're able to track the exosomes somehow, then you can start to study how this intercellular communication takes place. And this will be very important later on for whatever diagnostics or therapeutic applications we want for exosomes. And also, using micro-nanofluidics, we'll be able to isolate and identify the specific exosome subpopulations, as I was saying before. Okay. So some of the ways that people have been thinking about using micro and nanotechnologies for exosome isolation are microarrays and also microfluidic devices. So microarrays are some like subset of microfluidics. They're called microfluidics because you deal with small droplets. In this microarrays, basically you print like a set of different droplets on a substrate. These droplets have capture antibodies on them. This is like a close-up of this section. So you have a capture antibody on each of these droplets, and then you flow a sample through them. The sample, the antigens, will be captured by the capture antibodies. So whatever is in the sample that relates to this capture antibody will get attached here. And then afterwards, you do some sort of like an ELISA test. So you put a detection antibody, reacts to a different marker on the surface of your sample here, and then you get a fluorescence response, which you can measure with your fluorescence microscope. So this is one of the ways that people have found to isolate exosomes. And these are the particular like cocktail, they call them, of antibodies that they've used, for example, CD9, CD63, and CD81, which have been found to be specific to exosomes. So that with this kind of platforms, they're able to detect and also to phenotype exosomes based on what their combination of antigens on their surface are. Okay, so So this has the advantage of being able to capture them, to detect them, to phenotype them, but then it's hard to like release them, or maybe you want to know the contents of this, and then it's hard to lyse them. Further purification of the samples within the exosomes is a little bit hard. So this is good for certain applications, but then not so good for others. Okay, another application or another approach that people have found is use of microfluidics and also microbeads. So in this case, we have this work by He, and in which they have this microfluidic device. This is done on PDMS, you know PDMS and all the soft lithography. So they have this device with different inlets. They start to put the samples in. The first one, they put the exosome sample in, and the first step is that they have this previously mix the sample. For example, it could be plasma or urine or whatever. So they mix the sample with microbeads that are conjugated with an antibody. So it's basically like this, but instead of having the antibodies on a surface, you have the antibodies on a microbead. So the microbeads target the exosomes. They attach to them, and then since they are magnetic, they are arrested by a magnet. You put them here in this like chamber, in which then they flow a lysing solution to release the contents of the exosomes. So by this, you keep the beads here, you keep like the membrane of the exosomes attached to the beads, but you release the contents. And then afterwards, they use a different set of antibodies, like a detection antibody for the fluorescence, the chemifluorescence detection of the contents of the exosomes. So in this case, they were using this microfluidic device to analyze the contents of the exosomes. So it also works. You can use specific antibodies to try to detect certain particles, but as with the other example too, you have to be very sure about what you think you might find in the exosomes, because otherwise you select a set of antibodies that maybe don't target anything that's within your exosomes, and then you think there's nothing there, while in reality you were just not targeting the appropriate proteins or whatever you're trying to find. So you either have to use a lot of antibodies, do a lot of tests, and then see what you get inside, or you have to be very sure of what you're putting into your system. Okay, so finally, for this design challenge, your goal would be to work in teams, I think you already know what your teams are. They were sent to you by email, I guess. Otherwise, I have the next slide, I think, with the teams. So you work in teams throughout this week. There are some brainstorming sessions along the way. You get together, you think about this problem, and the objective will be to design a microfluidic platform for the analysis of exosomes coming from a single cell. So basically, what I want you to think about is how you could devise a platform in which you could separate cells into single cells and then analyze the exosomes that are being secreted by each of these cells. So by this, you can analyze one cell, get like a profile, for example, of the exosomes that are being secreted. Then you go to the next cell, do the same thing, whatever. And then by the end, when you have your population, you might see a trend, right? You analyze the cells, all the cells, and then you see a trend of this specific, I don't know, cancer cell. You get this exosome like release trend. So this will be helpful later on to compare this profile that you come up with a sample like from a real patient. If you find this profile, then you could be pretty sure that the patient has a cell that is, I don't know, cancerous, and it's releasing a certain type of profile that you already found before, right? So you will present all these designs on Friday. You're gonna have 10-minute presentations, and then there's gonna be space for 5-minute questions and answers within like all of you. So there are no like wrong or right answers in this design challenge. It's just to let you apply your, imagine how you would tackle this problem, and see that the tools that you will be learning throughout the week have an application and they can be very useful for real world problems. And so remember that these are very important. Exosomes are small, so they're very small. You have to take this into account when you're designing your platforms. So try to not rely on complex or expensive systems for every test. For example, you've seen before that it's possible to make nanometer structures using e-beam lithography, but if you have to use them for every test, then it gets very expensive. It gets very time consuming. So even if it's possible, try to not use this kind of systems for every test. If you could do like, we need e-beam lithography for just one wafer, and then afterwards we get a lot of devices from it, then it's okay, right? So you have to make an e-beam lithography wafer every time you want to run a test, then it gets very complex and expensive. So also remember that exosomes are heterogeneous. Even though some specific markers have been found, you cannot think that just using one antibody you will be able to capture all the exosomes because of this heterogeneity, and also because of the stochastic nature of exosomes. And finally, be creative and just come up with ideas, and it's just for try to have fun. Okay, so these are the teams. This will be online, of course. Probably you already know these teams. If you know the people, then you're lucky. Otherwise, you have to be social, communicate with these people, send emails, try to have lunch together, go for a coffee. Okay, so this will be the judging criteria. So first of all, just meeting the design challenges. So I think you have to be turning in advances or something like that. I don't know. You have to, okay. Yeah, basically you just come up with your designs, you try to make them feasible, you try to present them in a structured way, you try to engage the audience, try to make this, as real as possible, and don't go too sci-fi or think about maybe things would be cool, but maybe if it's not feasible right now, the judges will say like, I don't know. So it's very hard to say this is gonna work, or this is not gonna work, because a lot of things are not known on this topic. So it's hard to come up with something that you would say I'm sure it's gonna work, or something like that. But try to make it real. Like try to explain why you think certain applications would work, why certain techniques would work, or just propose something and try to back it up with information that you can get online or papers you read or something like that. And also include a process flow for your system. For example, if you're doing some microfabrication or soft lithography or something like that, well, include the process flow, how you would fabricate the device. Like what would be the photolithographic steps, or how would you do this? And also explain the merits of your designs, like why the system would be better than something that's being done right now, and how it will help to advance the science. And so told that there's going to be a first place prize, and I think it's going to be a gift card, right? So I think it's going to be a gift card, one for each of the team members. So I'll try to make it as good as possible, and you'll get something at the end. That's it for me. So do you have any questions?
Do you have an idea what order of magnitude your concentrations are for exosomes?
Concentrations, I'm not really sure. You have, for example, in a certain plasma sample, you could get like 3 million exosomes in a microliter. So if you work with real world samples, then you get a lot of exosomes because they're very small, right? So the concentration would depend on how good your system is and how good your purification is, but you would expect to have a lot of exosomes in a normal cell population.
I understand you wanted initially just to isolate cells.
Yeah.
And then see how many exosomes are, well, try to probe the exosomes that are exuded from that cell.
Yeah.
So that concentration is not the one that you find in a typical bodily fluid.
Yeah, it would be different, and also it's hard to tell the exact concentration because it will also depend on the time. Like you sit them for more time, then they will probably secrete more exosomes. And then some people say it's also possible to like make the cells secrete exosomes with particular signalings. So I think maybe pH changes or something like that can make cells secrete more exosomes in time. So I mean, I don't know an exact number of how many you would get, but I don't know, it's possible.
Well, if there are no more questions, well, first of all, we can thank Alex for the presentation. Now we've got some time here where you guys can get together, discuss, meet each other, know what your strengths and weaknesses are. If you're a mechanical engineer, talk to the biologist about what these things are, vice versa. And so you know, be free. And yeah.